In this lecture, we will discuss about cell wall structure, its chemical composition and functions. So we know that plant cell is always surrounded by a rigid and protective layer around plasma membrane. And the cell wall was first discovered by Robert Hooke. So cell wall is outside plasma membrane and is very rigid in structure and protective in function. So when Robert Hooke examined under compound microscope thin slices of cork cells, so actually he sees cell walls, the chambers, he see the honeycomb chambers like structure he sees was actually a cell wall. So he first discovered the cell wall. Cell wall is non-living structure which is formed from the protoplasmic living protoplasmic material, and cell wall is made after the completion of cell division during cytokinesis. A plant cell without its cell wall is called a protoplast. So when cell wall is absent, the cell is so actually a protoplast. And plant cell wall is made up of cellulose, hemicellulose, pectins, and proteins. So these are the chemicals which from which cell wall is made. In fungi, cell wall is formed from chitin, which is a polysaccharide. Bacteria contains protein lipid polysaccharide complexes, also known as peptidoglycan. So it is a mixture of protein lipids polysaccharides. So it is present in bacteria. Also, cell wall contains plasmodesmata. Plasmodesmata are pores present in the cell wall, and they help in communication between cells and transport of materials across cells. So these are present in cell wall. The pores present in cell wall is known as plasmodesmata. So this is in this structure you can see here. This is one cell, this is another cell, and you can see this yellow line. This is plasmodesmata. This is plasmodesmata, and you can see here this is cell wall cytoplasm, and the two pathway is given. This is vacuole apoplastic pathway, symplastic pathway. Apoplast pathway is when the transport occurs through cell walls. This is apoplastic, and when the transport occurs through cytoplasm, it is known as symplast pathway. The blue color is symplast pathway. So these are plasmodesmata which help to communicate between two cells, and also transport of material occurs. Structure of cell wall. Cell wall consists of three of the following structure. First one is primary cell wall. It is the first formed cell wall, and it is the outermost layer of the cell. So primary wall is the first cell wall formed, and it is the outermost layer of the cell wall. In immature meristematic and parenchymatous cells, it is the only cell wall. So in immature meristematic and parenchymatous cell, only primary cell wall is present. Primary cell wall is comparatively thin, permeable, and contains cellulose, hemicellulose, and pectins. So it is thin and it is permeable and contains cellulose, hemicellulose, and pectins. Some leaf and stems epidermal cell contains cutin waxes and make primary cell wall impermeable. So waxes and cutins are fatty compounds, fatty acid-like compounds which make the primary cell wall impermeable. Primary cell wall of yeast and fungi is composed of chitin, which is a type of polysaccharide. So it is primary cell wall. Next is metal lamella. Cells of plant tissues remain cemented together by an intercellular matrix known as the metal lamella. So metal lamella act as a cemented material which um, make two cells join in a plant tissues. So it act as a cemented material. Metal lamella composed of pectin, pectic acid, and some proteins. Secondary cell wall, it is present between primary cell wall and cell membrane. So this is between primary cell wall and cell membrane is secondary. This is a secondary layer, the second layer. It contains cellulose, lignin, glycoproteins, and secondary cell wall consists of three layers known as S1, S2, and S3. So you can see here in this structure, this is the transverse section of a plant cell. So you can see here, this is these three, this is the secondary wall, this is the S1, this is S2, S3, these three represent secondary wall. This one is primary wall, and this one is middle lamella. This is the longitudinal section of cell walls of two cells. You can see this, the middle is middle lamella. The middle lamella act as a cement to join two cells. This is one cell, this is another cell, this is primary wall, and this is secondary wall. You can see this is S1. The first one is S1, second S2, and S3. Chemical composition of cell wall. The cell wall contain different types of organic compounds. So we will discuss it in detail. First one is cellulose. Cellulose is a major component of cell wall. Cellulose composed of approximately 1,000 to 10,000 beta D glucose residues, and a key component of primary and secondary wall. So cellulose molecule is a polysaccharide which is made of monosaccharide glucose molecules, and this is the D glucose molecules is help to form cellulose. Cellulose polymer associate through hydrogen bonds, forming microfibers and macrofibers which interact with each other to form fibers. So different types of cellulose polymers associate with each other through hydrogen bonds, and these form microfibers, and microfibers associate with each other to form macrofibers, and macrofibers associate with each other to form fibers. Cellulose microfibrils wind together and form fine threads. They may coil around one another like strands in a cable. So microfibers wind together from one another and form a fine thread to form macrofiber, and they wind around one another to form a coil like strands in the cable. Cellulose framework of wall is interlocked by a cross-link matrix of hemicellulose pectins as well as glycoproteins. So the framework formed from cellulose is interlocked by different types of non-cellulosic compounds such as hemicellulose pectins and glycoproteins. So you can see here, this is a cellulose molecule. So this is glucose molecules combine to form cellulose. These are chains of cellulose. This is one cellulose, is second, is third, is fourth. So hydrogen bonds occur between individual cellulose molecules, and these form microfibrils. These are fibrils, and these microfibrils wind to one another to form macrofibril. You can see here, this is these fiber combine and make macrofiber. Macrofiber combine and form fiber. So this is the fiber, microfiber and microfiber, and this is the glucose molecules forming cellulose molecule. And this is the mixture of non-cellulosic molecule with cellulose. This is cellulose microfibrils, and these are non-cellulosic. This is pectins. These are non-neutral pectins molecule. These are acid pectins, and these are calcium bridges between pectin molecules. These are glycoproteins. So these various molecules cross-link with cellulose molecule. Next one is hemicellulose. It is a polysaccharide composed of a variety of sugar, monosaccharides, xylose, arabinose, and mannose. These are monosaccharides and carbohydrates. Hemicellulose molecules are often branch and hydrophilic. These are branch molecule and is water soluble. Abundant in the primary walls, so also found in the secondary wall. This is mostly in primary walls, but also occurs in secondary walls. It plays a significant role in regulating cell enlargement. So hemicellulose ensure to and regulate the proper enlargement of cell. Next is pectic acid. It is the major component of middle lamella, but also found in the primary walls. Pectic acid is a polymer of about 100 galactronic acid molecule, and it is hydrophilic in nature, that is water soluble. It is also carbohydrate. So you can see here, this is middle lamella, this is primary wall, this is plasma membrane. So this is protein, soluble protein. These big rods are cellulose microfibrils. These small fiber is pectin, you can see here. And this is hemicellulose, this greenish color. These are hemicellulose. So these are the different types of molecules which are found in the typical cell wall present in plant cell. Next one is pectins. It is a polymer of around 200 galactronic acid molecule found in primary walls layer and middle lamella. So pectin is a larger molecule than pectic acid. Compared to pectic acid, it is less hydrated, that it contains less OH group and highly hydrophobic, and this is more water soluble. Extensive calcium cross-linking of pectin occurs, preventing further stretching. So in the cell wall, cross-linking of calcium occur, which make a calcium bridges of pectins, which prevents further stretching of cell. So it also regulate proper stretching of cell. Next is glycoproteins. A structural protein such as extensins was originally presumed to be involved with cell wall extensibility, but the deposition of this extensin, now we know that deposition of extensin may actually strengthen cell wall. So glycoproteins are a structural protein, and one of the example of glycoprotein is extensins, which was originally involved in cell wall extensibility, that it gave an extension to cell wall. But now we know that the deposition of extensin may actually strengthen the cell wall. Next one is lignin. It is a glue-like substance deposited in woody plant cells, and because it provides strength, and the deposition of lignin is called lignification. So it gives strength to the woody plant cells. Callose, it is a polysaccharide composed of spirally wound chain of glucose residue, and the structure is spiral structure of glucose chain because it is a polysaccharide made of glucose associated with wall of sieve elements of angiosperm phloem, which transport food material, deposited rapidly in response to mechanical injury, environmental or pathogen-induced stress, and sealing the plasmodesmata, which is the cytoplasmic strand between two cells, contiguous cells. So callose, when a mechanical injury to cells occur or environmental or some pathogen attack on the cell, so it uh, releases and seal the plasmodesmata, which is the pores in the cell wall, and help to avoid that injury or in pathogen-induced stress. Callose is involved in pollen tube development and also transiently associated with cell plates of dividing cell. So also shortly help in cell plates formation. Next is cutin. It is a wax-like deposit found in the epidermal cell exposed surfaces, and it makes cell surfaces impermeable to water. The deposition of cutin is called cutinization. Cutin deposition is very thick in xerophytic plot because they are water deficient, so they need to regulate its water evaporation. Next is soberin. It is a pet-like deposit found in secondary wall of cork cells, and it makes cell wall impermeable to water. And it also regulates evaporation of water, and the deposition of soberin is called soberization. Both cutin and soberin occurs in combination with waxes and function largely to reduce water loss. Mineral silica is deposited in the cell wall of grasses. Calcium oxalate and calcium carbonate are found in the cell wall of Acanthaceae, Cucurbitaceae, and Moraceae families members. So these are also very important. Mucilage mucilage is mainly found in the seed coats in aquatic plants, which imbibe water and retain it for the long duration. So mucilage help in retaining water for the long duration. Next one is cell wall formation, that how cell wall form. So when cytokinesis start, cell wall is formed from Golgi complex endoplasmic reticulum elements which align on the equatorial plate or the central position of the cell. Next, then middle lamella is first formed by deposition of calcium and magnesium pectates. Next, primary wall is secreted by the protoplasm lies inside the middle lamella. From that, secondary wall is secreted by the protoplasm lies inside the primary wall. So cell wall formation occur in the when the cytokinesis starts, and it forms from Golgi complex endoplasmic reticulum elements. So you can see here, this is the telophase and this is the Golgi bodies and the endoplasmic reticulum elements here. You can see here, this is the cytokinesis. As the cytokinesis start, so the pragmoplast, these are the microtubule structure which help in cell plate formation. So you can see the vesicle from Golgi apparatus and endoplasmic reticulum associated one another and form a cell plate, and this is the new cell wall is formed. So this the division of cell occur. This is one cell, this is another cell. So first primary middle lamella, then primary wall, then secondary wall occur. So cell wall formation occur. Next is functions of cell wall. Cell wall play important role and it provides support to the plants. So cell wall provide mechanical strength and support to the plant. Also, it withstand turgor pressure. Turgor pressures is the force exerted against the cell wall as the contents of the cell push plasma membrane against the cell wall, and it helps a plant to remain rigid and erect. So cell wall is rigid and strong, so it withstand the turgor pressure, the force exerted by the internal contents of the cell, and which push the plasma membrane, and plasma membrane is pushed against the cell wall, and in turn it helps the plant to remain rigid and erect. Cell wall helps in regulation of growth. It sends signal to cell to enter the cell cycle. In the cell cycle, the cells divide and grow. Cell wall regulates diffusion because cell wall is porous, allowing some substances to pass into the cells. Communication, as we know that cell wall contains plasmodesmata and through which cell communicate one another with the help of plasmodesmata, which are pores between cell walls, and that allows molecule to pass between the individual cells. Next is protection. Cell wall provides a barrier to protect against plant viruses and other pathogens and help to prevent water loss. Storage. Cell wall also store carbohydrates for use in plant growth, especially in seeds. So this is all about cell walls. Thank you for watching.